Now, the neuron, which serves such important functions, is encased in three connective tissue sheaths. The innermost layer is called the endoneurium. The next layer is the perineurium, and the outermost layer is the epineurium. Now, as you can see in this diagram, the individual nerve fibers, they are covered by the endoneurium. Now, a group of nerve fibers is referred to as a nerve fascicle, and the fascicle is covered by the perineurium. So you have many nerve fascicles, and together they form the whole nerve. So the nerve as such is covered by the epineurium. So endoneurium, perineurium, and then the epineurium. Most of our nerves are myelinated. Myelin is very crucial when it comes to the speed of transmission. So let's look at the process of synthesis of myelin, which we refer to as myelinogenesis. Now, what is the myelin sheath? So the myelin sheath can be seen as a white segmented sheath which covers the axon of a myelinated nerve. Myelin serves many important functions. It protects the nerve fibers, it insulates them, and most importantly, it accounts for the speed of conduction. Now, myelin is essentially made up of proteins and lipids. The lipid portion is very important because this is the one which provides for insulation, and as we shall shortly see, this contributes to the high speed of conduction. Now, the process of formation of myelin differs between the central nervous system and the peripheral nervous system. In the central nervous system, the oligodendrocytes form myelin, whereas in the peripheral nervous system, the Schwann cells form myelin. Now, as you can see in this slide, in the central nervous system, there is an oligodendrocyte which myelinates many axons around it. And in the peripheral nervous system, it is the Schwann cell which myelinates a single axon. Now let's examine these processes in more detail. Now in the peripheral nervous system, it is the Schwann cell which forms myelin. One Schwann cell only myelinates one axon. So the Schwann cell rotates around the axon many times, and it lays down myelin in the process in several concentric layers. And the Schwann cell now begins to occupy the outermost layer, and the membrane of the Schwann cell forms the outermost layer of this entire coil, and that is referred to as the neurilemma. Let's look at the diagram that explains this. So as you can see in this diagram, a single Schwann cell myelinates only a single axon. So you can see how the Schwann cell itself rotates around the axon. And as it rotates, it lays down myelin in several concentric layers. And when it is done with the process, you can see that the outermost region is occupied by the Schwann cell. So the cell membrane of the Schwann cell is now forming the outermost layer, what we refer to as the neurilemma. So you have the axon, then the layers of myelin, the Schwann cell, and finally the neurilemma. But myelination in the central nervous system is different. So as we already saw, the cell that forms myelin in the central nervous system is the oligodendrocyte. And one oligodendrocyte myelinates many axons in the neighborhood. So the oligodendrocyte does not move in this case. It only sends out its processes to myelinate each axon. So the processes would lay down myelin around the axon. So unlike the Schwann cell, which itself rotates around the axon, the oligodendrocyte remains stationary. It only sends its processes. So one single oligodendrocyte myelinates many axons, which means that if there is loss of myelin, then the oligodendrocyte will not regenerate that myelin. Unlike in the peripheral nervous system, where if a neuron loses its myelin coat, then the Schwann cell again forms myelin around it. Now that we have studied the structure and function of the neuron, let's talk about the different types of neuroglia. As we already saw, the neuroglia are the supporting cells of the nervous system. Now, in the central nervous system, there are 4 different types of neuroglia. These are the astrocyte, the ependymal cells, the microglial cells or microglia, and the oligodendrocytes that we already saw. Now let's first talk about astrocytes. As the name itself suggests, these cells are star-shaped. Now you can see in this diagram that they are intimately associated with the capillaries of the central nervous system. So the astrocytes together with the endothelium of the capillaries form what is referred to as the blood-brain barrier, which is very important in filtering out undesirable substances, including microorganisms. So the function of astrocytes is to regulate extracellular fluid composition in the brain and of course form the blood-brain barrier. The ependymal cells line the ventricles of the brain and the central canal of the spinal cord. As you already know, the ventricles are fluid-filled cavities inside the brain. Now, this means that the main function of ependymal cells is to secrete cerebrospinal fluid. So whether it is the ventricles or the central canal, the fluid that fills them is the cerebrospinal fluid, which is secreted by the choroid plexus. So the choroid plexus is made up of ependymal cells. Now the microglia are actually the macrophages of the central nervous system. They are specialized macrophages. So their main function is phagocytosis. Oligodendrocytes, as we have already seen, form myelin in the central nervous system.